selection and continue giving off different forms of tumors? If you kill off 99% of one cancer, will it come back as exactly genetically the same cancer, or is it actually different?
That's a very sophisticated question, and I guess that a biologist may put a term on that some of you may have heard, which is clonal evolution. And so the question is, for example, if you have a cancer that's made up of different clones of cells, in other words, the way that we might see a cancer would be that there would be a hierarchy or an organization within the cancer, where there's a cell on top of the hierarchy that's responsible for initiating the cancer, but then there's a bunch of other cells that are responsible for maintaining the cancer. And then over time, as the cancer grows and changes, is it possible that one clone could be killed off by a certain therapy, but then another clone could rise up to take over the growth of the cancer? And I think the question is yes. The answer is rather yes, and I think that we have seen evidence in some cancers of clonal evolution, and that when some clones are killed off, others may take over. But the whole genetics question becomes even more complicated, because it's not only about a susceptibility by having one gene already mutated, and then predisposition by having the other allele of that gene or the partner to that gene also mutated, and then cancer declaring itself. There's another layer of things as well, and that's called epigenetics. We haven't brought that up yet, but as you can tell, the name implies above genetics. So those are heritable changes that happen in DNA and cells that are not a result of an altered DNA sequence. So in other words, it's something that can change the expression of a certain gene without actually having a mutation in the gene. So that's brought an additional level of complexity to the study of cancer. Because now we must not only look at the genome, we have to try to understand the epigenome, the things that happen above that level, and how those two things work together such that clone may recur. So I guess what I'm saying is there may be a genetic mutation that you target, and that's good, but then maybe the next clone is rising up because of an alteration in the epigenome. So now there's just another whole layer of things to confuse the question you're asking.
Hi. So my question is to those of you who are practicing clinicians as well as in the lab. To what extent is the process of moving research to the clinical side accelerating now? What are the kind of processes that are helping that happen, hindering that to happen?
So that's an amazing question, actually, and it's something that is concerning many of us. I've been involved with the National Institutes of Health in the United States to try and help address that question as it applies to cell therapy. It's a huge issue, and the reason is that research funding is based on discovery research. It's what's called hypothesis-driven research. And in order to move observations in the laboratory to the clinic, what one has to do once you've made that observation, that basic observation, is move it, translate it. That's part of what's called translational research. But then you come to a stage where it has to be tested, the treatment has to be tested in mice, and it then has to be tested not in humans, but in the system that you're investigating. That's particularly true for cell therapy, because it's a complicated system that requires many laboratory procedures before the cells are suitable for administration in patients. That research is not discovery research. That's validation. And that's extremely difficult to fund through conventional mechanisms. I think we're making progress, and I really welcome the opinion of my colleagues here. I think we're making progress in the States on that issue, because it's been a huge challenge. And what I can tell you is that with regard to cell therapy research, I was telling Steve this at the beginning of the evening, is that study that I described to you is exceedingly expensive. Now, we'll eventually be able to refine it, and it'll become quite a bit cheaper. But just to give you an idea, to treat one patient costs $90,000 to do that study. There are very, very few existing clinical trials, research funding mechanisms that can fund that. And there is increasing focus on how to do that, how to move things from the lab into the clinic. You can apply the same rigorous criteria, it's just that the criteria are different. You're not proving a hypothesis, you're validating an approach.
Well, I think, as Armand said, the NIH they probably invest more per capita in the U.S. than here, so there's really much more of an affirmative action approach to research than in Canada, although things have improved here, but I think they're still behind, or we're behind. They've actually tried to outline these tiers of research, and as Armand said, most of the money goes into discovery, but they're trying to shift the resources. And unfortunately, people say, well, it's up to the drug companies. And it is very much up to the drug companies, but during a recession period, the drug companies have to be very careful how they're spending their money too. So there's been a bit of a bottleneck lately, and people are trying to streamline the ethics process, the approval process, the regulatory process, the oversight process. But then, one bad trial, where someone dies in a gene therapy study, and then suddenly everything's kind of paralyzed for a year or two. So I think overall it's improving, but it's obviously slow. But in many cases, it's slow to protect the public.
I agree, and I think too the important thing is that in Canada, I think maybe the difference that Steve was alluding to between Canada and the States is they just have so much more funding overall to actually put drugs, new discoveries through the development and the proper phases to test toxicity and all of those things. For us, we make a basic discovery, and the question you asked, how do we translate those discoveries? Right now, our healthcare system just does not have the dollars to actually have the hospitals and the universities actually carrying out that research and development. And so what you often find is some critical, great key discovery is made, and then it's handed off to big pharma to develop it, to package it, and to then subsequently market it and sell it. And I find that in a lot of places in the States, that entire process can be rolled into one academic process that can be undertaken by an extraordinarily wealthy institute or university that happens to exist in the States. We just don't have that kind of funding. So we're in a position where there are some scientists that actually want to participate in that development and delivery to the public process, but are restricted by the fact that when you've made the discovery, it's just too much of a laborious and expensive process. So recently, a drug company came around with McMaster, whose name I can't divulge, but there was a drug company that came to and wanted all the scientists to come to a talk that they were giving. And what they were proposing to McMaster is that, and I'm sure that they've done their rounds around all the universities, they were proposing to McMaster that they wanted to set up a parallel laboratory next to our laboratories, whereby whatever new discovery we made, they'd be right there next door for us to literally hand it off. And I'm sorry, I don't know any football analogies, I'm not good at that, but and they kind of hand the ball off to somebody else. So when they do that handoff, then they're right there to catch the new discovery, and then they say, don't worry, we'll take care of all the work, and we'll take care of all the development. But the issue here is then you've introduced a corporate element. And I like to think, alluding to the question that happened before, that people, although this wasn't reflected in recent surveys, but I like to think people still trust their positions as being members of the public, and that we're part of publicly funded, government-funded institutions, and overall, I guess, we're not corporate. And so I'd like to think there's still some trust there. But then when you do hand the ball over to big companies, of course, there's money at issue, and there's money making an issue, and there's some corporate interests to discuss. So we need to, as Canadians, decide, what do we want our process to be, right? And is it really important? Do you want us sitting here to be the ones to carry the drug through to delivery to the patient so that it may be more meaningful that way, or should we continue with big pharma? And that's a huge question.
So maybe I can comment on the political process a little bit. I think that there's the beginning of an understanding of the impediments to moving basic research into the clinic. And my sense is that our leading research institution, which is the equivalent to NIH called CIHR, Canadian Institutes for Health Research, is very receptive now to the notion of supporting research that is going to move to the clinic. And there are other organizations that are specific. The ones I'm familiar with are multiple myeloma, Leukemia and Lymphoma Society, and so on, that are very interested in helping that process to take place. And the federal government, somewhat to my surprise, also appears to be interested in supporting that. But you know, none of this would be happening if we weren't being vocal about this. In a sense, cell therapy, if I may talk about cell therapy, is a little bit of an orphan area because it's difficult to make a business case for profit. And large pharmaceutical companies are grappling with this issue. That whole area is absolutely fascinating because what I've discovered as I've explored this is that the returns on drugs by big pharma have declined over the past decade. And they're actually continuing to decline. So they're now looking at other approaches. And one of those approaches was the use of targeted therapy in the form of an antibody, monoclonal antibodies. So it has taken 20 years to go from the lab into using monoclonal antibodies this morning for a patient with lymphoma. But it's now standard therapy and it prolongs their life. So it's really important. But 20 years ago, it was impossible to make a business case for it. And it's the same with cell therapy now. It's very difficult to say, well, this is how we're going to make a profit out of these messy things called cells.
I'm curious why you asked that. Is it from a business case point of view or a personal point of view?
Well, there's definitely an element of both. I'm first a scientist myself. I'm trained in biology and then engineering. And then secondly, I run a small healthcare startup specifically focusing on the cost cutting within hospitals and dynamic staffing because we're burning too much money too quickly to be sustainable.
This is curious.